Using MERFISH for spatially resolved transcriptomics at the single-cell level. Single-cell biology can provide important insight into the role of differential gene expression in cellular structure and function. One methodology is to sequence a cell's transcriptome, the set of all RNA molecules transcribed in a cell. However, single-cell RNA sequencing methods are error-prone and lack information on RNA spatial distribution. RNA location is important because the presence of RNA in particular sites can reveal mechanisms behind differentiation. For example, in this image we can see the movement of mRNA into the cytoplasm after transcription. Single molecule fluorescence in situ hybridization, or SMFISH, is a method for imaging RNAs using fluorescent probes that hybridize to transcripts of interest in fixed cells. However, current SMFISH methods that use super-resolution microscopy can only measure 10 to 30 mRNA species at a time. A new study suggests a methodological advancement called multiplexed error-robust FISH, or MERFISH, which can image thousands of distinct RNA species in live cells with low error rates. MERFISH can be understood through a simplified example. Suppose we're interested in locating 7 species of mRNA in a cell. We can identify each species with a unique 3-bit binary code, but misidentifying any one bit will change the code to that of another mRNA. To reduce error, we will use 6-bit binary codes and choose 7 of 63 possibilities. We will explain why this reduces error rates later. The next step is to create probes for each mRNA species. Each probe is a DNA oligonucleotide consisting of 20 to 30 base pairs. These probes utilize Watson-Crick base pairing, which is a result of unique hydrogen bonding patterns. Adenine forms 2 hydrogen bonds with thymine or uracil, and cytosine forms 3 hydrogen bonds with guanine. An oligonucleotide will hybridize, or bind, to the complementary sequence on its target mRNA once it has permeated the cell. We now conduct multiple rounds of hybridization. In each round, we will only include probes for species that have a 1 in the corresponding binary code position. For example, in the first round, we will include probes for species A, B, C, and D because they all have a 1 in their first bit. At the end of each round, we use conventional fluorescence microscopy to locate each probe. The probes are then photo-bleached and a new subset is added. We conduct 6 rounds because our codes are 6 bits long. Once the on and off patterns of fluorescence are converted into sequences of 1s and 0s, we can compare these sequences to the assigned sequences to determine the identity and location of each mRNA. You may have noticed an error during round 6. The on signal here was misidentified as a 0. By strategically assigning only 7 of the 63 available codes to our target mRNAs, we ensure any 2 binary codes differ by at least 3 bits. This significantly lowers the probability of misidentification. Because this code was only wrong by 1 bit, its identity can be determined by finding the closest assigned code. In order to validate MERFISH, the authors performed MERFISH measurements on 130 genes in human fibroblast cells. They used a 16-bit code with at least 4 differences between any 2 codes to encode 130 different RNA species. Because it is more likely to miss a hybridization event than to misidentify a background spot as mRNA, the authors limited the number of 1s in their assigned codes to further minimize misidentification rates. As shown in this graph, limiting the number of 1s and using 4-bit differences resulted in the lowest misidentification rate. Within the single cell depicted here, the authors detected more than 1,500 RNA molecules after error correction, which corresponded to 87% of the 130 encoded species. In addition, as shown in this graph, about 80% of fluorescent spots for each RNA species were decoded correctly. To validate the RNA copy numbers determined from MERFISH, the authors performed SMFISH measurements on 15 of the 130 genes. The authors graphed the copy number determined by SMFISH versus the copy number determined from MERFISH on a log scale. If the numbers match, we should expect to see a good fit to a y=x line, as shown. In the future, MERFISH will be valuable for examining gene expression in cells without removing them from tissue, which will be valuable in studying organs such as the brain. The spatial resolution of MERFISH may also be particularly important for understanding the function of long non-coding RNAs, which remain poorly understood. Ultimately, MERFISH will allow scientists to address new and interesting questions in developmental and system cell biology.